what I'm going to talk about is not just about cancer and about hereditary cancer, but about what we have found through our research about the Meitei community. And uh, if every community helps themselves, the whole nation and the whole world is helped really. So I think we say charity begins from home. So whenever I meet any part and I have such a large database, I know in which part of the country, which community, what are the common cancer risks. And uh, Reena just now mentioned about the cancer statistics of Manipur, and she said number one cancer is lung cancer. We know why men develop lung cancer. I'm not going to talk about that. I will talk about the most common cancer in Manipuri women, which is breast cancer, and also cancer, which is ovarian cancer, which is number three cancer. I will talk about that. And I will just share my one second. Hmm, just okay. Oh, can you see my slide?
Yes, sir.
Is it
Yes,
in
sir.
full screen? Yeah.
Yes, yes.
Okay. So, what I'm going to talk about inherited cancer risks due to founder mutations, and I will explain this term. What is a founder mutation? What is a BRCA gene? And of course, I don't have to tell you about Meitei community and what I know. Maybe I need to learn from you all that the Meitei community. I understand it's about 15 lakhs or 1.5 million Meitei speakers whose first language is Meitei, and of course in Manipur, and of course now you all are all over India and all over the world. So this is all I know, and this is all I knew. But now through my research, I also know more about the cancer risk, and that is what I'm going to talk about. So I'm showing this picture. This is a one of the tabloids in Mumbai, Sunday Mumbai Mirror, and these two ladies who you are seeing here, both of them had breast cancer, and both of them have been my patients, and they have told the story. And this, what you see above that, is the pedigree. Pedigree is when you draw a family tree. You trace. You start with one person. You ask about how many siblings they have, brothers or sisters, their children, their parents, brothers and sisters, grandparents. So we draw a family tree. So whenever someone has a cancer or we suspect, we want to see whether it is running in the family or not, we draw at least a three-generation family tree minimum. So one generation above and one generation below the affected generation. So this is what we do. And these two women, so when I was doing my, I think Reena mentioned I joined in Tata Hospital as a consultant in 96. Before that, I also did my MD from Tata Hospital in radiation oncology. So the senior lady, she herself is a gynecologist. I was treating her as a resident, breast cancer. And those days I had little idea what is. We were focused about, how to treat her breast cancer, etc. And later on, when I became a consultant, I got interested in genetics. And then I realized that she had a significant family history. We found a mutation in her, then her daughter. And we tested more than 30 members in the family, and many of them had cancer. And of course, we also prevented many cancers in the family. So this is just to give you a nutshell about hereditary cancers. And this is something known as hereditary breast ovarian cancer syndrome. And in short form, we call HBOC. And why we call breast ovarian cancer? Because in these families, the most common cancer in these families is breast cancer, and the second most common cancer in these families is ovarian cancer. And of course, you can get some more cancers. I will talk about it later. So, what about cancer genes and hereditary risk? I will explain to that. I believe many of you would not be from the biology or medical field, so I will try to make it a little simple. And since I've been given almost an hour, so I will, try to explain the terms. So, as you see here, is the cursor seen? My cursor when I'm moving the cursor?
Yes, yes.
Okay. So you see in the cell, body is made up of I don't know how many cells. Just imagine 1 milliliter ml of blood. When you do a blood test, it has 5 to 10,000 white blood cells. It has few lakhs platelets, and it has millions of red blood cells. Okay, that is the number of cells in just one drop of blood, not even one drop, 1 ml of blood. So that way, whole body is made up of cells, and cells, special type of cells, they come together to make a tissue, and different tissues make different organs. Like some will make our brain, some will make our blood system, others make, eye, some will make a, pituitary gland. Like that, there are different. So cells, tissues, and organs, and when all the organs are put together and they're functionally coordinated, then you have an organism. So human body is composed of that. So inside each cell, human cell, except red blood cells and platelets, which don't have a nucleus, there are 23 pair of chromosomes. And when I say pair, that means number 1 to 22. Each one of them, you get one from the mother, one from the father. Number 2 is also one from mother, one from father. And then there's a pair of sex chromosome, which is called X and Y. So females have XX chromosome and males have XY chromosome. So if the sperm from the father is carrying the X chromosome, mother egg only has X chromosome, a daughter is born XX chromosome. And if the sperm from the father, which fertilizes the egg, carrying the Y chromosome, then it becomes XY, and a son is born or a boy is born. So this is 23 pair of chromosomes. Okay. And chromosomes are made up of DNA. This is a special chemical of life called deoxyribonucleic acid. You would have heard of RNA. So this is DNA. And that through a process, I will explain to you a little later, DNA has the blueprint of all life forms. DNA has the blueprint. And from this DNA, through a process I will show later, protein is formed. So proteins actually do the function. They will make insulin, they will make our hair, they will make, decide our blood group, and they will make bones, everything else. So all enzymes, hormones, cartilage, bone, everything is made up of proteins. So this is how the body functions. So we are born with just 23 pair of chromosomes, and then when we are conceived, and as cells multiply, the same set of chromosomes and DNA are passed on to different cells. And as the fetus grows and different tissues are formed, some genes are expressed in some cells, some genes are expressed in other cells. And that's why different cells have different property and different tissues have different functional property. And that is how the body functions. That is how the whole system works. So now I will go about cancer. So this is just a brief. I'm not going to explain this in detail right now. I will come later again about this. Is if you see that I'm talking of the BRCA1, 2 genes, like that, there are many other genes. This is just one gene, common gene. So if you see in the human body where I've shown these dots, that means a cancer can develop. So when do you suspect a cancer could be hereditary? And when I'm saying is, everyone it's a dreaded word, cancer. No matter who you are, whether you're a doctor, you are young or old, no matter what your profession, you may be a soldier, but everyone is afraid of the word cancer because we have seen people suffer from cancer. How the movies are talked about cancer, someone has to die, they will say, okay, the hero or, or the heroine developed cancer, and that is how they die. So the word cancer instills fear in us, and anyone who has cancer, they always think, why did I get cancer? Of course, someone who has been a heavy smoker and develops lung cancer, they do not ask this question. They know the question, and everyone else around them. But anyone else who develops cancer asks this question: what did I do? Did I eat something? Is it because of pollution? Is it because of? But one of the reason for cancer is genes. About 5 to 10% of all cancers are because of genetic, or they have inherited the risk. When they were born with a change in the gene which increase their risk for cancer. So these are known as hereditary because it is from the genes, it is genetic, and anything it is in the genes which is passed on to the next generation, it becomes hereditary. So briefly, when someone has a family history of cancer, or there are some other features also I will describe later, even in absence of family history, we suspect this person's cancer could be hereditary. And if we suspect it is hereditary, the suspicion alone doesn't help. Like if we suspect someone has cancer, we have to do a biopsy to confirm. Same way, if we suspect someone's cancer is hereditary, then we do a test to look if those genes have the alteration. I will explain later. And before that, there's genetic counseling, then genetic testing, and then we find a mutation. And I will explain later how we use this information. First, let me talk about what is DNA, what are genes, what are genetic variants, what are traits, and what are disease risks. Each of these terms. So, DNA, I have already mentioned, I will describe in detail. Genes, I will talk about. Genetic variants, again, so traits, all of us know, in some family, most people are tall. So, it's a trait. In some family, many people are very athletic, not just families, many tribes, races. So they have certain traits. And how do these traits come? Because of the genetic variants which are more common in that particular family or in that tribe or in that community or in that country. Okay. Like you see in people from the Afro-Caribbean, they have very tight curly hairs. So it's a trait. It's not a disease. It's just a variation. It's a trait. And that is because of the genes which they have. And some of these things that they have, the genetic variants, does not just give them a trait, but increases the risk of disease, which I will talk about it later. So gene is not just any stretch of gene is the functional unit of inheritance. So they have, as you see that it's a chromosome, pair of chromosomes. So chromosome is like basically, and it's a thread of DNA double helix. So you would say very famous double helix, DNA double helix. So they are sort of tightly wrapped around nucleosome and so very tightly packed, and then it becomes a chromosome. And I said they're a pair of chromosomes, one you get from mother, one from father. This is how it is shown. And genes have parts. One is exon, which makes the protein, and there's intron in between. So they have a specific structure. Not just any stretch of DNA is a gene, but specific structure of the DNA. We call it a gene. And gene basically directs synthesis of a specific polypeptide chain, which we call protein. So polypeptide means multiple amino acids. You have heard of amino acids. So the genes have triplet codons. They code for specific amino acids, and it's a chain of amino acids, what we call a protein. So human genome, which has 3 billion bases. Bases are in the DNA. So there are four letters: A, T, G, C, adenosine, cytosine, thymine, guanine. So 3 billion times it is written in different combination. Like the English language, we have 26 alphabets. The same 26 alphabets, the sequence in which to use these alphabets, you can write anything. Shakespeare has written so many books just using those 26 alphabets. Everything is written by the 26 alphabets. In the way you use and where you stop and where you start and how you combine those alphabet, what comes before that. So same way, this of the 3 billion bases, about 2% of these 3 billion bases make genes which code for a protein. So the remaining 98% is not coding for a gene or a protein. We used to earlier call it as a junk DNA, but nature will not carry 98% junk. So they also have some roles. I will not get into those aspects in terms of regulating the gene function or other functions which we do not know. So when I say 3 billion, 3 billion is in one set of chromosomes. So it is 3 billion plus 3 billion, 6 billion. Sometimes you have to think, someone suddenly asks you how many zeros in a billion. So 1 million has 6 zeros and 1 billion has 9 zeros, after 9 zeros. So what happens, this I will just show you. So gene is a specific structure. So this is like a DNA. So in DNA, there are exons and introns. Okay. Exons, if you compare it like the bogies of a train, and introns are the parts of in between. And finally, the protein is made up of the the DNA which is in the exons. So there is a mRNA and there is RNA, and the RNA is a process of transcription. In the transcription, the intron, the parts of DNA in between the exons are removed, and then they join together and mature mRNA is formed, and that forms a protein. And protein, like the DNA has a double helix structure, protein has a very defined structure. Each protein is different. That's why it functions differently. Otherwise, and genes, I did not mention. We humans have little over 20,000 genes. One may ask why we need so many genes, because human body is very complex. From the time human life is conceptualized, so from that till the time we die, in different organs, different tissues, different genes are expressed, they make different proteins or they are silenced. Okay. So mind you, whether it is a brain cell or whether it's a white blood cells or whether it's cell in the liver, all of them have the same 20,000 genes, but they are expressed in different genes are expressed. When they say expressed, that means the proteins that they form. And once a protein is formed, then that protein also makes some changes happen to the protein, what we call post-translational modification. So today I will focus about in terms of mutation is what happens when there is an alteration in a gene, which is the DNA, that affects the protein which is formed. And if the protein, because that's the final molecule which works, then it will not work. And depends on what is the function of that protein or that gene, a disease related to that could develop. This is more scientific talk. I will not talk about what regulates the expression of the gene. I will not go into details. So for cancer, there are two types of genes. One is tumor suppressor genes. So they suppress the natural tendency of some cells to become cancerous. So that's why they're called tumor suppressor gene. And then there are oncogenes. So they are normally growth factors. So if there is something wrong with them, the mutation, then they can accelerate the development of cancer. Then these two words I have written somatic and germline. Germline, that means the germ cells, you germinate like a seed germinates. So mother's ova or eggs and father's sperm, these are the two germ cells. When they come together, when they fuse, then a zygote is formed, then embryo is formed, they multiply, one cell, two cells, four cells, eight cells, 16 cells. So this is germline. Anything which come from our parents, as a germ cell, it will be present in each and every cell of our body because our life started with that one cell and that piece of DNA. So it will be present in every cell of the body. The other thing I've written somatic. So somatic means in the tissue. Sometimes it is not the germ line, but during our lifetime, in some cells of the body, in one organ, some DNA is changed because of whatever reason, because of the ionizing radiation, because of some carcinogen, because of a mutagen, or just because of the aging process, DNA could change. And that is happening only in those cells. It is not there in all the cells. So that is known as somatic change. Now, there is also, I've talked about this, a genetic change is epigenetic. So epigenetic, because after gene has to express, so there are certain things which could affect the way the gene is expressed into a protein. So it is not genetic, but epigenetic. So there are also changes sometimes that can also cause some disease and other risks. Adult onset diseases can happen because of epigenetic changes. So today we will focus in our talk about germline genetic changes, and BRCA1 is a tumor suppressor gene. Okay, it is not an oncogene, it's a tumor suppressor gene. So what is the ultimately as doctors, this is a diagnostic test. Why doctors want to find out if someone has cancer as to what is there in this thing. So somatic NGS is a technique known as next generation sequencing. Sequencing means you try to find out what is the sequence of DNA in this sample, whether it is a blood derived DNA, which is germline, or from the tumor derived DNA for the genes of your interest. So when we find some alteration in some genes in the tumor cells or a somatic mutation, it helps us to decide precision treatment. See, normally the drug treatments like chemotherapy, cytotoxic chemotherapy or radiotherapy, they don't distinguish. They will just go and hit whatever it is. And it so happens that tumor cells are more sensitive to these cytotoxic agents like chemotherapy, drugs, or radiation. They are killed more. Plus, the normal cells, even if they are killed, they have a greater ability to repair. But in precision treatment, you're not just sort of killing it at random. You are finding the weakness in the tumor by some genetic alteration and you're targeting that. That is known as targeted therapy. And one more treatment is immunotherapy. So in some mutations, immunotherapy works, and the approach of synthetic lethality. Like someone who has a mutation in a BRCA1 gene. Now, BRCA1, I will just talk about BRCA1. So BR stands for breast, CA stands for cancer. And this was the first gene which was found to cause hereditary breast ovary cancer. That's why it is known as BRCA1. BRCA2 is the second gene which was found. That's why it was called as BRCA2. And the normal function of BRCA gene is to repair any damage that happens in the DNA. So I mentioned there are 3 billion bases in human genome. So imagine someone is copying, someone gives you a task, one page, you type it again and again, or not just copy paste. Okay, you have to actually type it or you handwrite it. No matter how good you are, if you type it 100 times or 200 times you write those pages, you will make some error. Okay, of course, nature is much smarter and that has machinery, but in nature also, for every million bases it copies, because once it is copied, from one generation to other generation and then cells multiply. For every million bases, one error happens. So that is a baseline mutation or a variation rate that happens. Of course, there are mechanisms to correct it. And plus, as we live, because of various agents, there are the carcinogens or mutagens which cause mutation. So DNA could be damaged. So BRCA1, BRCA2, and some other genes, the proteins which they make, they are there in each and every cell. Whenever a DNA is damaged, they sense, they have a machinery, they sense, oh, here the DNA is damaged in this part. They will go there, they will call some other friend, some other proteins, and they repair it. That is a normal function. So if in a tumor or if in a person, a BRCA gene itself is mutated, the person who was given the job of repairing this itself is defective, then those tumor cells are susceptible. So if you block the other way of repairing the DNA, damaged DNA, then those cells die, the cancer cells die. So this is known as synthetic lethality. So you make it the treatment more lethal. Now that's also somatic. And if you find a germline mutation, that means that is when you do on the DNA which you isolate from white blood cells or any other normal tissue. And again, for them, you can do next gen sequencing or other methods like Sanger sequencing. And if you find a mutation, it helps in precision treatment, which I explained, and precision prevention. People in this family, everyone is worried, will I also get cancer? Instead of everyone worrying, as I showed in that pedigree, you can find out who has inherited the cancer risk and who has not inherited. And then based on that, you can do that. And cascade testing. So cascade testing is a cascade, it falls from there. Suppose person A, Sunil has a mutation in BRCA1 gene. So his children would have a 50-50 chance. So how does this come 50-50 chance? Because each gene is in a pair, as I mentioned earlier. BRCA1 gene is on chromosome number 17. So Sunil would have two copies of chromosome 17 and so one copy would have the mutation, other is normal. So half of his sperms would have the mutated copy. So when these gametes are formed, sperm or ova is formed, then the cell splits into half. So half of them would have the BRCA1 mutation, half of them would not have the BRCA1 mutation. And depending which sperm goes on to find for fertilization, a child is born. Each child would have a 50-50 chance of inheriting the mutation, not inheriting the mutation. So instead of guessing, we can check who has the mutation, not. And suppose Sunil's son has or daughter has the mutation, then in future her children would also have a 50-50 chance of inheriting the mutation. But if suppose Sunil's daughter has not inherited the mutation, in future her children would not have the chance of inheriting the mutation because she doesn't have the mutation unless until from her husband's side there is a mutation coming in that. Okay, so this is the utility of doing finding genetic alteration. I am talking about cancer genes, but there are many other genes, there are many other hereditary conditions, similarly like thalassemia or many other conditions like that. So what happens, what we are interested in, one is like chromosomal, so types of genetic alterations which could happen is the whole chromosome, like, Down syndrome, you would have, heard of. There are many other syndromes in which the entire chromosome, it is either duplicated or removed. So that is numerical, or it sort of goes and breaks. Like there's a disease called chronic myeloid leukemia. In that there is a BCR-able translocation. So one part of one chromosome goes and joins the other part of other chromosomes. That is a translocation. So whenever a whole chromosome is multiplied or deleted or numbered, then there will certainly be some effect on that. Then parts of the gene are removed or doubled, duplicated, then again, it depends on the function of the gene, it would have an effect. Then whatever it is in green, that means it is unlikely to cause effect. Whatever is in red, that means likely to cause an effect. When you have a deletion, that means many bases, nucleotide bases are removed. And I will explain to you the mutation that we have found in the Meitei community. They are frame shift mutations. So this is a reading frame, how it is read because from exon, it becomes pre-mRNA, and then this becomes a mature RNA. It reads, okay, where then there's a stop codon. Remember there's a stop codon, it terminates that no further, it will be terminated. This because ultimately the DNA stretch is a continuous, but there is a specific sequence of DNA. It says stop now, no more protein will be made. It will be only 1863 amino acids in BRCA1. But if suppose that sequence which makes the stop codon, it is introduced earlier, instead of 1863 amino acids, the BRCA1 protein will be much short. It will have only 120 amino acids, say 400 amino acids. So large parts of the protein which is required for its function for DNA repair are absent. And that is why, so what we call truncated protein, and that will pathogenic. That means it will cause the pathology which is associated with this tumor suppressor genes. And here it is breast cancer. And then there are other type of mutations. I will not go into this because there are various ways a mutation can cause. Now, mentioned there are triplet codons. Triplet codons have 4 bases, A, T, G, C, and they can form 64 possible combination, and but only 20 amino acids are there in humans, 20 amino acids, and one of the coding is for stop codon, and there's one is a start codon. So something which will there the DNA, if it reads that sequence, you say okay, start. Okay, you start making protein, and then stop, you can stop making protein. So it is a code which is read through that proteins are formed. So what happens is basically, I said DNA, genes, amino acids, and the proteins have biological function, and mainly it is for normal growth and physiology. So because of any reasons, if there is a change, whether it is spontaneous reason or because of a mutagen, you have an altered DNA sequence. And previously we used to call it any alteration in DNA sequence technically is a mutation or a genetic alteration. And so you would have a gene with a genetic alteration or a mutation. But mind you, as I mentioned earlier, only some DNA alterations in a gene will change the structure or shape of the protein which is coded by that gene. Okay, so in many times you would have a DNA alteration, but it is not causing any change on the protein, and proteins are the effector molecules, so nothing happens. And many times, even if the protein is altered, but the biological function remains the same. So it allows for that much change in the protein, and the biological function remains the same. And sometimes that is a marvel of human physiology, that even if the protein is changed or the biological function is affected, but there are other mechanisms to take care of that. But sometimes that some biological function changes will cause a disease. It could be congenital anomaly or evolution. But because only some of these changes are causing this effect, and many times they are not causing, there's something known as variant of uncertain significance. That means we find an alteration or a rare alteration in a gene about which we do not know that it will cause the disease related to this or not, or it is a normal variation. So when we say normal variation, that means in that population, we know that this happens maybe in 1 in few hundred people, it's a normal variation. Based on all this, we classify when we do a genetic, basically it will just read the sequence. And based on that, what we find, we call it either a pathogenic or a likely pathogenic variant. And there's the American College of Medical Genetics, we use a classification. So we know that in this gene, if this DNA change is there, there's a very high chance that it will cause a disease related to this gene, genetic disease. And benign or likely benign, that we know this is a variation in this gene, but because of the type of variation and it is not so rare in the healthy population database, we know that this is just a normal variation. This will not cause a disease. And whenever we can't fit into any of these, we call a variant of uncertain significance. So now this question comes, why do you have variation? See, if you see someone from Afro-Caribbean, without even looking at their genetic or genotype, you can tell them this person is of African descent. When you see a Japanese, when you see an Eskimo, when you see a North Indian, when you see a Meitei, you just by looking at them, their traits, you can see and how it has happened. Like an Eskimo would have thousands of places where their DNA is little different from, or at that particular position, it is different from someone else, let us say from Ethiopia. And someone from, let us say a Jat from Haryana, he would have another thousands of places where in his DNA there will be changes which are different from, say, someone else in Japan. Same way, in your Meitei community, there will be thousands of places where there are DNA alterations which are different from, say, anyone else, say from Assam or from Bengal or in any part of the India. That's why different diseases. And how does this happen? Because people have married or have children through people from their own community. So whatever the alterations happen, remember I said 1 per million bases, and we have 3 billion bases, they keep altering, and those alterations are passed on to next generation, and they accumulate and they give rise to this. So how did we know the pattern of inheritance? So this is Gregor Mendel was an Australian monk. Okay, he was actually a botanist and geneticist, but he was monk. That was his, I think, profession. So he did a lot of experiments with pea plants. I think all of us would have studied this in school or college. I don't know which class they teach it now. And through he looked at various traits. I'm talking about traits like, say, hair of Afro-Caribbeans or anything else, skin color. Many of these are all traits. He looked at different traits of the pea plant and the seeds. And through that, he deduced that there are some dominant traits and there are some recessive traits. Okay. And what he found that, and that is true for human genes also, some alteration that only one copy. Remember, I said there are two copies of every gene or alteration when you get from mother or father. There are certain things where only one copy has the change that itself will manifest or express. So it is dominant. And recessive, when both copies of that is affected, then only you will get the effect of phenotypic effect or a trait that is recessive trait. Okay, and there are many human conditions or diseases which are recessive, like for example albinism. You would have seen an albino who have no pigment in their skin, in their eye, in their hair. So it is autosomal recessive. Both the parents will look absolutely normal because they have alteration or mutation only one copy of the gene which causes albinism. But if they have a child, 1 in 4 chance, the child will inherit both mutated copy and then only it will manifest. So talking about the hereditary cancers. So hereditary cancers, because it is germline mutation, if you remember, we have found with, and each gene has a pair of, each gene is in a pair. First hit means a mutation. In all cells of the body at birth, they have the first hit. Whereas someone who does not have, who has not inherited any mutation from the parents, all the cells in his or her body would have no mutation in that gene at birth. So what happens, as I said, again mentioned that mutations are probabilistic events, 1 in a million probability. So it is just a matter of time that some cells of that person's body will develop a second hit. The same gene would have another mutation. And once both copies of that gene are mutated, that means no functional copy is remaining of that gene, then they would develop cancer in that. And because these people have a bond with the first hit, they have a very high lifetime risk of cancer. Otherwise, normal say chances of developing breast cancer in women, in Indian women, is around 3 to 4%. 1 out of 25 women will develop breast cancer. But if someone is born with a mutation in BRCA1, their lifetime risk of breast cancer is around 80%. Okay, so the risk becomes very high. And normally, if a woman develops breast cancer, mostly it will be in one breast only. But if someone has mutation in all cells of the body, they have a 40% chance of developing cancer in both the breast. And because that mutation is there in other cells of the body also, they are also at risk of developing ovarian cancer. So plus, because they are driven by a particular genetic pathway, here where example is of BRCA1. So their tumor, they're a type of breast cancer known as triple negative breast cancer. That means that these are three receptors, estrogen, progesterone, and HER2, all are missing. So it's a typical type. And plus ovarian cancer, the different types, there's something known as high grade serous ovarian cancer. They are more common in those who have a BRCA1 mutation. I am focusing more on BRCA1 because that is a founder mutation in the Meitei community. Okay, so because they already had a lead time bias, they will develop cancer at an earlier age. They would have family history. They could be bilateral or multicentric. So this is what also the scientist known as Alfred Knudsen, he explained this in a childhood cancer known as retinoblastoma. But this, what is explained in the retinoblastoma is applicable for all hereditary cancers. So now I will explain how this information is used. So when we suspect someone has possible hereditary cancer because of family history, if no family history, then either early onset cancer, like say breast cancer typically happens in 50s, 60s or 70s, but if someone got breast cancer at their 32 or ovarian cancer at their 35, so it is much earlier than the typical age for that cancer. Or bilateral, that means develops cancer in both breasts or breast and ovary. So we suspect that this person's cancer may be of the hereditary type. Then we have the counseling and then do genetic testing. And if we do find a mutation, I will not talk about uncertain significance or no mutation because there also could be polygenic or like a diabetes. If we find a mutation for BRCA1 gene, let us talk about, then we do the counseling for the risk management. So as I mentioned that there are targeted therapies and chemotherapy. Those who have a mutation in the BRCA1 gene, there are many chemotherapy drugs. One is cisplatin or carboplatin. They exploit the weakness in the DNA of the tumor cells because they have BRCA mutation. They can't repair the DNA damage, so they work better. And a targeted therapy, there's a known as PARP inhibitors. So their double strand DNA break ability is already inhibited because of the BRCA mutation. The PARP inhibitors also inhibit their single strand DNA repair. So single strand and double strand, both strands of DNA are damaged versus single strand is only one strand is damaged. So that is what helps in the treatment. So if we know in breast cancer, if we do this, the chances of cure are improved by 10 to 15%. Ovarian cancer, again another cancer, if we give PARP inhibitors, the chances of cure are improved. Suppose for that stage 3 ovarian cancer, the chances of cure was only about say 50% long-term cure. If you give this targeted therapy, then the chances are increased by another 10 to 15%. Okay, so that is how it helps. Okay, so that is for the treatment point of view. Then early detection. So otherwise, just knowing that I am at risk, some people know that I have a family history or it may happen without family history also. I mean, you say be careful, but I mean, how would you be careful? You do maybe once or twice, you do scan or you check up. After that, you forget, and then the cancer develops. So there's screening or surveillance. So we know for which gene, which cancers at what age is a risk, whether you are a male or female. And I'm primarily talking about females, but males are also at increased risk for some cancers. And that you do screening for that. So like mammography for breast cancer screening, the three ways was first you make aware them for females that you are higher than the normal risk of breast cancer. So just being awareness itself can sometimes help that if they have any lump, they don't just sort of shy away and not go to doctor. They should consult a doctor and get a proper evaluation. Then once in 6 months, a doctor who knows how to examine breast, they should examine. And plus, uh, then screening with a mammogram. So mammogram or before the age of 40 or 50, mammogram may not show the cancer very well. So you could do a MRI of the breast. Normally, no one asks for MRI of the breast. So the screening to detect early. Again, I had to ask my patient, how many stages of cancer? Most will give it wrong. Everyone says 4 stages. So there are 5 stages of cancer. There's something known as stage 0 also. So in stage 0 cancer, where the cells have become cancers, but it is still within the lining of the duct. So breast is made up of milk glands. Milk glands have ducts through which the milk comes at the nipple, through which the child suckles the milk. So when the cells have become cancers, but within the lining of the duct, it is stage 0 or ductal carcinoma in situ, which is a Latin word for being inside. In stage 0, the risk to life is 0, just small surgery and plus whatever treatment. If the breast is removed, then absolutely 0 risk to life. Otherwise, again, it can be completely cured. Stage 1, the risk is there, but very little risk is there. As stage increases, the risk to life and the intensity of treatment increases. So screening, then prevention. So now I talked about breast cancer, and I've shown this diagram. Okay, if you see this for breast cancer, you have one option was for screening. The other option is do preventive mastectomy. So Angelina Jolie, the Hollywood actress, she shocked the world about 10, 15 years ago. She wrote her own story in New York Times. I think one of these, I forgot. So her editorial that she did preventive double mastectomy and of course plastic surgery to repair the breast. All the breast tissue which was inside, so the skin and nipple was left like that, but the tissue which you see, the milk glands and the duct in which the cancer develops, that was removed. And because it becomes hollow, either through an implant or by taking tissues from the tummy region or from the back, it was reconstructed by a plastic surgery. So you women who are at high risk of developing breast cancer, they have an option of screening for detecting cancer early, or they have an option of, by doing preventive double mastectomy with or without plastic surgery. For ovarian cancer, screening test, which is by an ultrasound or by a blood test known as CA125 tumor marker, it can detect ovarian cancer, but the problem is very often it will miss stage 1 or stage 0 ovarian cancer. I mean, so it doesn't really help. It just gives you a false reassurance that nothing is happening, but ovarian cancer is developing. That's why ovarian cancer is more dangerous than breast cancer, because invariably it is diagnosed in stage 3 or stage 4. So for ovarian cancer, we say that after the age of 35 or 40, once the family is completed, we remove both the ovaries and the fallopian tubes. Okay, because the cancer could start at the end of fallopian tube, although we call it ovarian cancer. So once that is done, it could almost eliminate the risk of ovarian cancer. Not 0%, but 1 to 2% it remains. So that is what could be done for this. Plus medicines like tamoxifen and some other hormonal agents, if we give for some mutation like BRCA2 gene, it can reduce the risk for breast cancer. So this is in a nutshell how we use the information. I've shown it, for most of you are lay persons from this point of view. So with this. So now we are coming about the population genetics and other mutation results that we have found. So we are 1.6 billion South Asians. 1.6 billion South Asians. We are there. Just one second. So we constitute 1/4 of the world's population. 1.6 billion. And you see that if how is the Himalayas, they decide, because this was a landmass which came from African continent, came and rammed into the Asian landmass, created Himalayas. Himalayas created all that, and majority of the population in the this is a subcontinent in the South Asian subcontinent, Indian subcontinent, has come from this pass from the west. But there are also some passes on the east. So many of in the northeast states, the Seven Sisters, a lot of population has come through the passes in the eastern end of Himalayas. And these two populations have not mixed again because they were isolated within that. So in South Asians, we have 1.6 million, 15 to 18 lakhs new cancers every year. And if you just say 2 to 3% of being hereditary, then you have 50,000 families who would have hereditary cancers, and everyone would have a brother or a sister or a child or parents. So you have 4 people. So you imagine we require so many people who require genetic counseling, genetic testing. But what do we have really? Ours, Tata Hospital, is the only comprehensive cancer genetics clinic. There are very few hospitals which have started. Now they have started, and that is there. And but it is required. So oncologist, and I'm very happy whenever I meet an oncologist, I tell them that at least you should know what is important aspects. You cannot become an expert in cancer genetics. Whatever is required for your patients, that much you should know. And I'm very happy one of our oncologists, Dr. Meitei, she's also joined, and I used her help to counsel a Meitei family because they understood only Meitei and language, and she helped me. And more and more people, doctors from the community, have to help. So what happens? Genetically predisposed individuals, like those who are born with a germline mutations, they are eminently suited for early detection and prevention because they have a very high lifetime risk of cancer. Like I explained in BRCA1, 80% risk of breast cancer, half of them would have cancer in both breast, 40% risk of ovarian cancer. And if a man has this mutation, then they have 15% risk of uh, they have 50% risk of prostate cancer and 2 to 5% risk of male breast cancer. Plus, both females and males have 2 to 5% risk of pancreatic cancer and some other cancers. So, by doing the testing, we can identify if they are at risk and you can prevent or the cancer or detect it early in morbidity. But what happens in low and middle income countries? Although these people have seen cancer in their family, they often present in late stages. Only some lucky ones are referred for genetic counseling or testing. And forget about before they develop cancer, even after they develop cancer, very few of them have genetic testing is done. And if they have a mutation, we do not know what proportion of them will develop cancer. And then there's the issue of cost, access, and compliance. Cost means how much it costs. Typically, genetic testing, the way it is done, it will cost 15 to 25,000. And whether it is done in, of course, in Tata Hospital, we can do cheaper for poor patients. Access, that means even if they can pay, but it is not available, genetic counseling and genetic testing, and compliance for genetic testing. Someone has explained to them and they do it. And even if a mutation is found, then screening if they are found to have a mutation and preventive surgery. And why I've highlighted all these things, because in the end, when I will ask you all in the question answer session that what is the utility? See, we do today in 2025, I don't do genetics only for curiosity. Okay. The day is long gone where people do genetics out of curiosity. Okay. It is for utility for that patient and for the family. And again, I will come to the next slide as to how it will help by finding a founder mutation and establishing it as population frequency. So how we started, I started, as I said, in 96 when I joined as a consultant. It was a registry. My, lunchtime, I used to see them because I spent a lot of time taking family history. Then 2003, we started once a week clinic. Then 2007, a daily cancer genetics clinic. Then I was in ACTREC. Through my ICMR, Indian Council of Medical Research, I established a lab. And then now we do provide very high throughput, extensive germline testing. And so I now also signed the report. And now we have more than 18,000 families, of which about 100 odd families are Meitei families. And I will talk about them later. So what happens in the process of genetic counseling and testing is that first thing is you do pre-test counseling, as I explained what we do in pre-test counseling. And inform them of the various option testing, and then we explain to them what is done. So these are the principles of genetic counseling: autonomy, empathy, beneficence. So I won't go into the details of these principles of genetic counseling, but the way medicine is practiced, most of the doctors we have trained in very busy hospital and the OPDs are crowded. Many times we don't follow, and it's a cultural thing also. I mean, we think that everything can be told to everyone, but genetic testing, genetic test status, sometimes the daughter doesn't want her mother to know at that point of time. She says, I will tell my mother when I think it is appropriate for me. So these are breached. So this is a little survey that we did in our patients. What we found that, of course, two-thirds or three-fourth quarters of the mothers who had breast cancer, they thought that I may pass the risk to my daughter. They were guilty. They were concerned about their daughter's marriage. And one-third was certain that because they have developed cancer, their daughter will also develop cancer. What was surprising, one-third of these women had not disclosed amongst their family members also, sometimes close friends or even their sisters also. And that I did not understand initially. How come you cannot tell your sister? Just imagine that lady tells her sister will tell her husband will tell his mother will tell her family. And because people marry within the community, it's matter of few years, there will be hundreds of families who would know in this family cancer is running. And because cancer has a taboo, no one wants this sort of disease. So that could be the reason. So this is the ethical, legal, and social implication. Myself started with this myth that in India everything is one large family, everything is shared. So this is really a myth. There is each person and each family is unique. You have to address their individual concern. That's why we require genetic counseling. You can't just say, okay, all women are like this, all old women are like this, all Maitis are like this, all Jats are like this. Each person is different. So I'll give an example of the society. This is a family which comes to me. It's a 2006 picture. That time I have all black hairs. So they come from the suburbs, from a village in Mumbai. They said they knew everything about cancer. One they said, you come and give a talk in our village. I said, why to your village? They said, sir, we know about cancer, everything. We know whether you have to come in time, then how you will treat. If you come in time, you'll be cured. But what we can't bear when our children come back from school crying because other parents tell their children, don't play with children from this family because there are a lot of cancer in this family, you may also get cancer. So the society needs to be sensitized that it is not by eating together or playing together or sitting together, it is a vertical transmission. It is through the genes which are passed on. Okay. So this is again 2003 when I started. So Myriad Genetics, they had a patent on BRCA genes, so that everyone had to do their testing. It was costing $3,000 in those days. So it is very expensive test. Who could afford it? So I spent. So this is also the land of Mormons. So Mormons is a sect. It's a Christian, one of the Christian sect. So they believe in family values. They will travel together. They have 6, 7, 8 generation family history tree they make. And this ideal for genealogical research. This is in Utah, Salt Lake City they have. So there I met these people. This is me, not be recognizable. And these are genetic counselors. So genetic counselors are the link. They are the bridge between patient, families, doctors, laboratories, and the society. Because it is very important that all are connected together, and I will talk about it later. A genetic test report could be misinterpreted. This has happened in US, not in some village in India. Washington Post. And he said, damage for rest of my life. Woman says surgeon mistakenly removed her breast and uterus. So what happened? The nurse misread her BRCA genetic test result as positive. Positive means that it is good, and the surgeon thought she meant it is mutation and they removed it. Okay. So doctors don't want to get into this trouble. So most doctors, if given a chance, they would not get into the genetic test report. And plus, what we call is the classical breast ovarian cancer is that if someone has a mutation, classical is that they get cancer at a very early age, triple negative type, in both breast, strong family history. But this is the uncommon presentation because you may have incomplete family history or unreliable family history or a small family also here also. Then incomplete penetrance. Not everyone, even every female who has the BRCA mutation will not develop cancer. 20-30% will not. And half the time it is coming from father side, etc., etc. Plus there are many other genes. In our own data, now we have 8,500 families. Breast and ovary is the most common cancer. And this is for pan-India cohort. We have done genetic testing for these 8,500. I will just show you the results. Depending on age at which they got cancer and whether they have family histories there or not, whether it is breast cancer, ovary cancer, the chances of mutation in BRCA1, 2 or some other genes is high. And ovary cancer, if you see, even in absence of family history, a high proportion of them, in absence of family history or age, they have a high proportion of them have a germline BRCA mutation. So this is our own data. What are the chances of? So from BRCA1 till the age of 20, if you're a female, I'm talking about whether you have BRCA1 mutation or not, doesn't matter. There's no, after that, cancer risk increases very rapidly between the age of 30 and 50, but even after that, there is increased risk. For ovarian cancer, the risk starts little later in life from say mid 30s and then goes up to 40%. So the French philosopher Voltaire, what he said, perfect is the enemy of the good. Now, what do you mean? All of us want to be perfectionists. We teach our children to be perfectionists. We teachers want to have their students perfectionists. So what it means in terms of genetic testing, that perfect system is you have a well-developed referral system. You do a pre-test counseling by a genetic counselor, then comprehensive NGS testing, post-test counseling, do the best you can do for screening and the treatment. So that doesn't really happen in reality. Even in our hospital, that is not being done for every patient. And what is good, at least the doctors who treat these patients, they can identify patient at a defined risk of cancer, like high-grade serous ovarian cancer or triple negative, or anyone with a breast cancer or family history. And if they don't have access to a genetic counselor, at least the doctors know how to take the basic, make basic pedigree, what are the common questions. And if you can't do a full gene, large panel testing NGS, then at least the hotspot or a founder mutation, again, I'll come to that, and use methods that can prevent cancer. So this is much older slide when I had not started looking at the Meitei community. So BRCA1 gene has 1863 amino acids and more than 10,000 bases, okay, the nucleotide bases. The mutation can be present anywhere there. So this says how many families had that specific mutation. So most common mutation in the Western India and Southern India is known as the dot 6869 del AG or 185 del AG, and there are many other mutations. So different mutations are more common in different communities, and that's when I will come to the Meitei community. So what is the uh, you know this thing? So this is, I found it very interesting website, Helix. So DNA consideration for space travel and colonization. What does it mean? So give you imaginary scenario. So imagine that 20 people were sent to establish a human colony in Mars, because you will not send 20,000 people, and only some few dozen people will go. And from these people, a new society would have to be formed over a period of 10 generations. But what kind of society would it be? So isolating a small batch of people from a larger population could have a dramatic effect on their society. It could be a cultural or a physical traits may be overrepresented. Maybe they were all very considered endurance because that's why they were sent to there. So all people who would be born, 80% would have that physical trait, or they were averse to music or something else. Many of the things that we have are also cultural. Okay, sorry, genetic basis of all the traits. So it could have, and this is concept that has molded population throughout history, because people of different races, tribes, communities look different, and this is because of the founder effect, because it started with some people, and that particular those people, 100, 200, 500 people, they founded that population. So one thing could happen, just imagine general population, they are different. There are, say, let us say 100 variations are there in the gene. And at some point of time, there's one isolated population. All the humans came out of Africa, and then from there they went all over. And so there could be two types of isolation. One is geographical isolation, that is either someone is an island, let us say Iceland or Great Britain, or there's a hill in between, large Himalayas are in between, or they are in a valley, or they are forests, or many of the central India for they are forests or deserts. And there's a cultural isolation. So they are living with many other things, but that particular, like in India in the last 1500 years or so, people had cultural isolation within, they would marry within their community. Although there were other people, they were not across the hill or across the in a different island, but they were marrying within each other. And there's something known as population bottleneck. That means a catastrophe may lead to a large drop in a geographically or a culturally isolated population. So suppose, so this example is given for the Ashkenazi Jews. So it is thought that once, because of whatever reason, it could be a war, it could be a natural calamity, their population reduced to say, just say only 100 people were remaining. Okay. And then this, but they still married within each other and had progeny. If out of those 100, two people had that particular mutation after say 100 generation, those two individuals with that mutation, today there may be 10,000 people carrying that same mutation. So the surviving individuals increase in number over centuries and dominate the future gene pool of that tribe or community. So when few people are separated from the larger population, some of the traits are disproportionately either represented or lost relative to the larger population from where they came from. That's why, people in different islands, would look different. And Darwin did his evolution theory. That is how he did. So different human subpopulations. I mean, we're all one human race, but races, tribes, linguistic groups, caste, etc. we have developed unique traits, and some of these carry increased risk of certain disease relative to each other. So the traits are not only athletic, or your hemoglobin level is higher, or you have a sickle cell trait, but sometimes also for some other diseases. Give an example of Iceland. Iceland is, I don't know any one of you have been to Iceland. I have never been. One day I want to go there. So what happened, in this diagram, you see three countries. So 1100 years ago, people emigrated to Iceland from Scandinavia, Scotland, and Ireland. So since then, very little immigration to the island. So today the population is around just over 3 lakh people, and they are mostly descended of this small group of ancestors. So some of the ancestors who initially came to Iceland more than 1,000 years ago, they carried a particular mutation in the BRCA2 gene, and that has been passed on, and today it has become very common. So the few people who were there, the founders today, because there more many of them, so one out of every 250 Icelanders is carrying this particular BRCA2 mutation. So and this could also, a higher percentage of Icelanders may be at an increased risk of developing breast and ovarian cancer because this gene mutation is common in their community. So this, I am showing this in the diagram of India. I have not shown the Meitei community. We have found that there are many mutations which are, I have written like 631 inst, that means 631 position insertion of T, or 484 del AG or 185 del AG. They are particularly common in particular regions and communities. If the mutation is very old, it will not be confined to one small region or one community, because as humans migrate, they carry the genes with them. But if it is a few hundred years old mutation, then that will be there. So we have all parts of this thing, like mutation that we find in Punjabis in India is also reported from Pakistan. There's a mutation in Nepalis 2214, and there in Biharis, in this all communities. So now I will come to, so when we look at the mutation, there's a method known as haplotyping. Through that we identify the mutation, that we find out that they had a common ancestor. So these are different mutations, a footprint of founder mutation. So now when we find a mutation, we try to find out how common is this mutation. So it is there in this community, but is it present in 1 in 10,000 people of this community, 1 in 300 people, or 1 in 1 million people of this community? If it is present in 1 in 1 million, obviously it is of no use. It's a very rare this thing. But if it is present every 300, 1 in 300 people, then you can help that population. So in Bombay, this is a community known as Konkanas Brahmin. We went to their society. Then we went in Bombay, all communities are represented. I'm sure there are many Meiteis also. This is a Syrian Christian church, to look at the mutation that we find in the Syrian Christian community. Father was very helpful. We got a lot of samples. So now coming to the Meitei community, the main point which I wanted to say that. So in the last few years, because we record in genetics clinic, there not just the state, but the caste, tribe, community. So from our records, I could find 75 cancer patients from Manipur and who had genetic testing, and 69 of them were Meiteis, and others were Kukis or Zo and some other communities and some Assamese, but 69. So I'm talking because each of them are very distinct, completely distinct gene pool. So what we found, and these patients had all either breast or ovarian cancer, and some other cancers also they had. So 56 of these patients had breast or ovarian cancer. Of these 69 Meiteis who came to us, 56 had breast or ovarian cancer, and in whom we found mutation in 23 of them. And all but one were the BRCA1 mutation, which I will explain later. So what we find is that this is, of course, patients who are coming to Tata Hospital, there's a referral bias, everything else. I'm not saying that there are 100 Meiteis who have breast or ovarian cancer, you will find mutation in so many of them. They're like 23 out of 56, because there's some referral bias. But this indicates a very high proportion of them, because in other communities, when they're referred for genetic testing, I don't find germline mutation in such a high proportion of cases. And what is important is that 21 out of 24 mutation that we have found in the Meitei community, there are two specific mutation at specific points that are there. So this can happen, and these are all unrelated families. Remember, we take 3 or 4 generation pedigree, so they do not recall this thing. And earlier we used to just write from where Impal. Now we actually write from which part of Manipur state they are. And so this is because that this itself suggests that maybe few hundred years ago, maybe 1,000 years ago, I do not know when the Manipuri Meitei community became isolated. The historians or your community historians will know it better. But at some point of time, someone developed this particular mutation in exon 15. Someone individual developed in exon 4 this particular mutation. And this third one is also probably a Meitei founder mutation because we have found in two and nowhere else we have found. And most of these mutations are not present in any population databases or disease databases. So these are founder mutation. So now my question is that I will come to that later. So we have to go to the public to educate them. Before the Angelina Jolie story came, we talked about it, and all the Asian countries are like us. The very little across Asia, very little awareness and genetic testing is there. We do a lot of genetic counseling training and for counseling and this thing. And we just last year we did all this was I'm talking of BRCA, but there are many other genes we talk about. And I now have 12 gene apostles with me. Gene apostles in the genetic counselor whom we are training, and we are starting a genetic course ADM. So I will so a lot of work. I will now come to the question answers related to my last slide. So we have been very generously funded by ICMR, DAE, DBT, and many others, a lot of people in this. Now I will come to my this slide for the question answer session. So the question is how finding a founder mutation, which we have already found in the Meitei community, and establishing its prevalence can help the community who has this founder mutation in a cancer predisposition gene. So I'll just put the two BRCA mutation that is there. So I want the discussion around this. Of course, anything else which you want me to clarify, I would like to have a discussion. Thank you very much.
Thank you, Professor Sarin. It was an excellent talk, starting from the basics of how our human genome, the genetic makeup, evolved and how it is processed inside our body and how the changes in the genome leads to the development and progression of cancer. And focusing particularly on the population specific cancer prevalence and how the gene mutation is related with that. We are focused on the Meitei population of Manipur. It is really exciting and eye-opening, particularly to those people who are not from the biological sciences background. So indeed, I think this talk is very helpful for it to everyone. So we can go forward with the question and answer session. So maybe Professor Sarin may start with this one, the question that you have posted. So it can be discussed around.
So before that, are there any general questions or queries if people can ask before we get into focused? If someone has very general, any chat,
Yes,
anything
yes,
else?
yes. I will put forward some few questions. Most of these questions are related with like the risk factor, the prevention, the awareness, and the diagnosis that is currently available. So one of the questions that I will pick up is like I will not individually pick up the questions from the individual scientists, but I'll merge it as a single question because some of the questions are more or less related to each other. So the question that I'm picking up forward is related with diagnosis. So the question is put up by Dr. Brajakishore Singh, who is a scientist working at the DBT IBSD Institute in Imphal, Manipur. And the question that he poses is, what is the easiest way to identify the BRCA1 and BRCA2 mutations? And in addition to that, I will add one more, like when one can start testing for genetic screening and how early we can detect it.
Okay.
Professor, yeah.
Yeah. So the questions are how to test for them and when to test. Okay. So when to test, I will address that question first is that, whenever you are at risk of developing the disease, because I shown in one of the slide, the cancer risk starts in females. The cancer risk starts, if you can see this slide, after the age of 20. So there's no point testing a 5-year-old girl because even when, of course, you will know whether the mutation is there or not, but even if the mutation is there, you won't do anything for that because the mother will get unnecessarily worried and panic. So when the risk starts, then you start testing, okay, from 20s. And if a male has the mutation, in men, the cancer risk starts even later, around 30s, the BRCA1 mutation. So maybe a little later you can delay. So this is the age when you do this. Now, how should you test? So if you remember, the genes can be, mutation could be anywhere. The mutations could be anywhere. If you see this slide, so this is a stretch of DNA. Mutation could be anywhere. To find that, and it could not be only just in BRCA gene. There are many other syndromes also which can have the gene mutation, which can cause breast cancer, ovarian cancer. So we test a panel of genes, what we call multi-gene panel, and we test maybe 15, 20, 30 genes by a method known as next generation sequencing. And because we do that, the cost of that is increased. Okay, it costs anywhere from 15 to 25,000 rupees. Plus, it's a special technique. So every lab, your local medical college lab and other labs, they will not be able to do. So most people send samples to private labs, commercial labs, and everyone is charged so much. But how that comes to finding the mutation? So suppose this is I will just show the mutation at a particular, I'll just use one of the slides to show where the mutation is. Okay. Okay. If you can see this slide, these are mutation at a specific points. If like in my research, the limited number of around 60 odd Meitei families who had with the BRCA, with the breast or ovarian cancer, in 95% of them, the mutation was at two specific points. Okay. And only in one gene, BRCA1 gene. I had tested them all for all the 26 or 84 genes, the entire region, but it boils down to just two specific points. So it is possible that if we have a founder mutation, or which is very common in that community, instead, if it is not possible to do full sequencing of all the genes by NGS, you can just test these two specific points. And that becomes much easier and cheaper. You can do it for 2,000 rupees, 3,000 rupees. And that is the whole purpose. I can help train people in Imphal or wherever else is there. They can find this mutation by very simple methods. Okay. So like if suppose you have to go in a hotel and you have to find some suspect somewhere else, you will spend so much time. But if I tell you the suspect will be either in one of these two rooms in the Almirah there, your job to find that will become much easier. So same way, if you know the founder mutation, we can just directly go there and check that mutation by Sanger sequencing, or even simpler technique, what by PCR we could check, and at least screening of the population could happen.
Yeah, continuing to that, we have one more question from Dr. Rajnikant, who is working as a scientist in the Wadia Institute of Himalayan Geology, Dehradun. So it is in continuation, which I think is a good question regarding the socioeconomic background of the people in the particular population. So what he says is, if a poor or middle class family wants to test both for the breast cancer and ovarian cancer, what is the best economic way to proceed? And it would be great if you have recommend a couple of ideas, including the treatment, and if there is any insurance or term plan covering that. So it's a
So
general question. Yeah.
as I mentioned, because we have found these two founder mutation at these specific spots, which are accounting for vast majority of germline mutation in the Meitei community with breast or ovarian cancer, the testing becomes very simple. And my aim would be, I would like to interact with the scientists and the doctors and people who run labs in Manipur and help develop these tests so that the state government can do this, the medical college can do this for anyone who has breast, ovary, prostate, or pancreatic cancer at a very affordable cost, and which the state can also provide.
Yeah. Thank you, Professor Sarin. Next, I will put take a question which is related with our this one, how aware prevention and related with lifestyle. So, what does our lifestyle, including the food and exercise habit as well as the age, play alongside genetic factors in cancer development and prevention?
So lifestyle as one of the factors we know for different cancers, different lifestyles are there. So like for lung cancer, different lifestyle risk factors. For breast cancer, it is related to the hormonal changes and obesity and maybe to some extent diet. So for different cancers, different lifestyle factors are there. The best lifestyle is to have your lifestyle of your grandmother, but with that is not possible. Okay. So in modern day, because of change in lifestyle, the cancer risk is increasing in general, but not to that extent. So one is a population risk. This I'm talking of high risk.
Indeed, that's definitely a big question in not only in cancer, in any of the non-communicable, metabolic related diseases, where lifestyle plays a very important role. And the next question I will take forward is the question which is related with why a population is particularly specifically prone to certain cancer, and why India, being having a high survival rate in cancer, the incidence of breast cancer is very high in India. So these two questions were put forward by two of the faculty from Manipur University, Dr. Himika Salam and Dr. Thangjam Davis. Professor Sarin.
So in different regions, depending on their lifestyle, diet is very different. In the entire Northeast, in different parts of the Northeast, in different tribes, the diet is very different. The carcinogens, which they are very fond of, are also very different. So accordingly, the cancer risks rates are different. Okay, so like in Nagaland, it is different. In Shillong, in the Khasis, it is different, and the type of cancer they are at risk is also different. So the focus of my talk is not that. That is very well established. A lot of studies, papers. So my talk is about what we have found is that one particular gene mutation, founder mutation in Meitei, is probably explaining a lot of breast and ovarian cancer. And this is a low hanging fruit. See, you asking the whole community or a tribe changes lifestyle or diet, you can try for that. It may take ages. Okay. It may take a generation, and by that time, the next generation would have taken up something else, which is maybe worse than the previous generation. Okay. But this is a low hanging fruit. We can identify who's at risk and we can, deal with it.
Yeah, certainly. Yes, definitely. This question, which I will put forward from my side, like we know there, apart from the genetic factor, we have lots of other risks, include what we have discussed right now, the right now, like about the lifestyle habit. So the question that I'm throwing to Professor Sarin is it's about what to say. A newly developed concept, as we call it, as a second the unknown organ of our human being. The microbiota that we have. So it is related with the diet, the lifestyle, and the host genetics also. So since we are talking about the ovarian cancer and the breast cancer, so the question I would like to throw is we have this reproductive tract microbiota, the bacteria, the microbes that are living
That's
in
true.
our reproductive tract in women,
Yeah.
like the vaginal microbiota,
Yeah.
the
Yeah.
uterine microbiota.
Yeah.
So, and we know that for ovarian cancer, human papillomavirus is one of the prominent causative agent of the ovarian cancer. So how
That is
does
for
the
cervical cancer. My HPV is for cervical.
yeah, yeah,
So your question is that how does the microbiome?
yes, yes,
So
yes.
the amount, the type of bacteria, the good bacteria and the microbes that we have in our gut, in our skin, in our all parts of our body, they could influence. They can either protect us from certain diseases. Cancer is one of the diseases, and that is there, and that the microbiome that we carry, our friend, the bacteria and microbes that we carry, is partly because how we sort of what we eat and what is our genetic makeup and the environment in which we live. So this is a very big area for research, and in India, especially in Northeast, many other places, the kind of food is very different, the fermented food and different each part of Northeast. Sometimes people here say, oh, Northeast is one thing. Northeast is not just one thing. There's so much diversity within Northeast. So
Yes.
that is beyond the scope of my talk, but it is certainly a very important. And ICMR, DBT, and some other organizations are actually doing research on this.
Yeah. So before I run off with the question, I would ask. I'll put a general question like when we go for a particular person or an individual go for genetic testing of the cancer. So let's say a person is identified with the mutations, which puts him or her puts her in the higher risk category of having the cancer. So
Mhm.
how would that particular individual will take forward her life? Like what are the things that need to be considered?
Okay. So that person, depending on the age, whether they are men or women who is having this mutation, whether they have completed family or not, whether they have had cancer or not, we advise them about screening, when to start screening, what kind of screening test they should do, and which I explained during my talk. And for breast cancer, screening is an option. For ovarian cancer, if they have completed their family, suppose a 40-year-old lady whose sister died of ovarian cancer and now has the same mutation in BRCA1, and she has completed her family, we may, for breast cancer, I may give her an option of continuing screening for breast, mammogram or MRI. And for ovary, I would strongly recommend that you remove both the ovaries and fallopian tubes, and that could be done either by open surgery or laparoscopic. So that will reduce her risk of ovarian cancer from 40% to just 1%. So that could be a lifesaver for her. And plus her children also. Suppose her daughter is 21, she needs to be tested whether she has inherited or not.
Yeah. So as of now, we do not have further questions. So we still have a couple of minutes to before we close the session. So while the rest other audiences put forward any other questions if they have, can we go forward with the questions that you have displayed, Professor Sarin?
Yes. So can
Yeah.
you see the question? Okay.
Yes. Clearly.
So
Yeah.
what is important is that how common. So first of all, I know that this mutation is common in the Meitei community, but I do not know how common it is. Okay. So whether it is present 1 in 10,000 Meiteis, or whether it is present in every 200, 1 in 200. So establishing population frequency is very important. And my starting point is people who have cancer. Of course, we do them, but to know, to estimate the population frequency, let us say 500 Maitis who do not have cancer or irrespective of personal or family history of cancer. And if we test them for these two specific mutation, and if we find that in out of, suppose we test 1,000 people and we find this mutation in say 20, that means 2% of the general Meitei population, or 1 out of 50 has this mutation. Then it is worthwhile screening. Of course, you will not say that everyone should be tested, but you would encourage people to get tested. And because telling them that in case they have the mutation, then cancer can be prevented in them and in their family members. That is one thing. The second is I am asking the question, how does it help? So what's the establishing the frequency of that mutation in that population? Like the Ashkenazi Jewish population, there are few mutations which are very common. 1 to 2% of the Ashkenazi Jewish population have that mutation. So people know that irrespective of family history, they ask in the hospital, do you have Jewish ancestry? They said just directly that mutation would be tested. Same way, if anyone who says that they are from Meitei community, they can be tested for these two mutations very easily at much less cost, and that can be done locally. You don't have to send the samples to Bangalore or to Mumbai and spend 20-25,000. It will save money, it will save life, and it will be done much faster because you know what to look for.
Professor Sarin, I am
Yeah,
Dr. Braskishore.
yeah,
Uh, in our office, it is Institute of Bioresource and Sustainable Development under Department of Biotechnology. We have sequencer in Imphal. So we can
okay.
sequence our blood samples and easily
Easily.
identify.
So what kind of sequencer you have? DNA capillary sequencer? What?
I think we have NGS sequencer.
NGS also. So for
Yeah.
this, you don't need NGS. This can be done very simply. Okay, your district hospital can also do. So we will talk about it later. So I would
Okay.
request the organizers to put me in touch with people,
Sure.
and that could be done. And for my own information, how many Maitis will be there in Mumbai? Few hundred. I just want to know.
Yes, yes.
Rina?
Definitely. Yes, definitely. It will be.
Sir, it is more than 100.
More than 100. Okay. So,
Even in Kharghar itself, around more than 50 people's Meitei
okay.
community. Some
Okay.
are from the Assam and some are
Okay.
from Tripura
Okay.
and
Yeah.
some
Yeah.
are from the Manipur.
Yeah. Okay.
Okay, then we can start from the Mumbai
Yeah, the
itself.
screening. Yeah. So main thing is establishing population frequency, whether this is of public health utility or not. That we will know only when we know the population frequency. But this is with counseling that in case a mutation is found, they should need not panic. And we when we work with the community, there's a specific method how we go about it and how to approach it. But it can be a lifesaver.
Yeah, Professor Sarin, I have one or two questions popping up right now. So it's regarding though it may not be under your domain. So it's a question which I think it's coming out of curiosity. Like everybody know that if cancer is there, we go for treatment, and most probably we go for chemotherapy and radiotherapy. So the question that is coming is there any possibility of a combination therapy, especially for breast cancer with cisplatin? Yeah, it's a drug I think used for cancer treatment.
Yeah, so there is a combination therapy for cisplatin, especially those who have a gene mutation in BRCA1 mutation. So cisplatin therapy works better for them. So it improves the cure rate.
Okay.
That is.
And this is a question from one of our audience whose mother died of cancer, but it's not either ovarian cancer or the breast cancer. It's the liver cancer. So she is asking is what is the chance that the percentage of risk that she can also suffer cancer?
So, many times we call liver cancer or bone cancer. Actually, it is a spread, say, we say. So if it purely
Professor
liver,
Sarin, your voice is breaking up. Yeah.
is it? Is my
Still
voice
breaking up.
breaking?
Yeah, now it's okay.
Truly liver, maybe the chance, the genetic type is less. Many times it is cancer or ovary cancer, which is liver and liver cancer. So in that case, it is many times ovary cancer. What happens? They have ascites or the tummy becomes swollen with water. So that is what people remember, and this is got ascites. So someone doesn't have a history of ovarian cancer, but they said my mother or my sister or my aunt used to have a lot of water in the tummy and they has to be removed. That could be ovarian cancer. And if that person is not alive for genetic testing, then their children or siblings, brothers or sisters can directly go for genetic testing.
Yeah. So we don't have further questions, and we still have maybe around 5 minutes more before we move to the next session, next agenda of the program. And I think if anyone is having anything that can be discussed that you would like to discuss, you can click or open your mic and can.
Someone has raised hands. Few people have raised hands.
Yes. Yeah. Dr.
I'm
Arun
Dr.
Moino.
Arun from PJ Chandigarh, Additional Professor PJ Chandigarh.
Okay.
So regarding the breast cancer.
Oh, you are from the physics, medical physics, no?
Yeah, I'm medical physics.
Yeah.
So in one of my discussion with a clinician, so they told that there is the increase of the breast cancer is happen with lactation. Those who do not do the
So these
lactating.
are, yeah, so these are general risk factors. Women who do not have children, or their first childbirth is at a later age, or they did not breastfeed. So in those people, the risk of, you can say, breast cancer risk is increased by 40%, but 40% of the baseline population risk. So baseline population risk is 3%. So it might become from 3% becomes 4%. Okay. So the risk is increased, but slightly. But if someone has a BRCA mutation, then that 3% risk goes up to 80%. So that's the difference.
So with the say nowadays most of the people they married in a very
Yeah,
old age
see,
period.
you can't change the society. Okay, no one
Yeah.
can live their grandmother's lifestyle. So that is why, in general, say maybe around 40 in villages of India, the breast cancer risk is 1/3 of what is the risk in the cities. Okay. And
Yeah.
so that you cannot change that kind of lifestyle. It is impossible to go back to the village lifestyle of your grandmother. But what I'm talking about is a really high risk.
So how much will be the high risk with the such type of the those who do not?
So the risk increases by around 40%. So if the
40%.
baseline risk was 3%, it will increase to 4%, 4.2%.
So who do not do the this lactating?
Yeah. Yeah.
Yeah,
Okay,
Dr.
thank you.
Kogan. Yeah, Dr. Kogan.
Yeah, hello.
Yeah. Hello.
Yeah, sir, it's a wonderful talk, and I'm not from a biology background, but it's really interesting talk. And the point that you made that like there is one particular area where this mutation strikes that you have
Yeah.
found through your
Yeah.
research. I
Yeah.
think it's a very interesting point where we can start with. But my question is like, if let's say if someone wants to do this screening testing or whatever you can call it, but
Yeah.
whether it how do we know it's available in the hospitals that we have in our state, or we
So
have to come all the way?
right now it is not available anywhere. So what we do, people send our samples, all their family members, like we have tested, they send the blood sample by courier. DNA is very stable molecule. By routine courier, you just send 3 ml blood and we test for this mutation. And because we know the specific mutation, the cost is also not much. It is just 2 to 3,000 rupees, 3,000 rupees to test for this specific Meitei mutations.
So I think what you mean is even I can also send my samples for my family, even though we don't have any history of
So, because
this.
otherwise, in absence of family history, we do not recommend genetic testing. But because this mutation
Okay.
seems to be quite common in the Meitei community, if you are from the Meitei community, then maybe it is worth looking at whether you have inherited either someone in the family has inherited or not.
Okay. Thank you, sir.
Yeah, it's Irina.
Sir, I have one question, sir.
Yeah.
Oh,
Yes.
Your talk is very enlightening. We
Thank you.
like very insightful also. So, sir, supposing if we have the BRCA1 and 2 mutation as being a Meitei lady, so what are the preventive measures once I know that is the mutation is being already happened, and then what are the preventive measures which I can
So,
take up?
yeah, so for BRCA1 screening, for breast cancer screening can be started. And we also look at the family history. Sometimes that also modulates the risk. And for ovary, preventive, no, removal of ovaries after the age of 35 or 40, once the family is completed. And that more or less takes away the risk, 98% chance that lady would never develop breast cancer or ovarian cancer.
yeah, it's Irina. Yeah.
Sorry, when I say
Uh,
98% chance, not for breast and ovary cancer, you are 98, 99%. Breast cancer, even if it develops by screening, we will pick it up very early stage and completely curable stage. Unless until someone is doing preventive mastectomy, which is a very major operation that I can't explain there. That is only if someone is very keen after counseling that is suggested. Otherwise, screening.
Thank you, sir. Thank you very much.
Thank you very much, sir, for the talk. And I want to ask one question, like the genetic counselor. So I want to know how that what is the qualification required to be a genetic counselor, and what is the like training, how the person can take training for that
So,
genetic counselor.
so that person should have done masters in some part of life sciences, masters. And then after that, so there is one masters in genetic counseling, also one or two universities offer. But otherwise, masters in any part of it with the experience. And Tata Hospital, we have started 1 year postgraduate diploma in cancer genetic counseling. So we have 12 people. So we train them in all those aspects. And if anyone is, so this is a good career option also. It also gives good work-life balance. You talk to patients, you talk to families, and it is very helpful. You know that I would love if someone from the Meitei community becomes a genetic counselor. They could help. This time I was lucky. Ma'am Tombi was there. I called her. She could help guide that family. But when she goes, whom will I call? I'll call you, Reena, to talk to that family.
Okay, sir. And sir, another thing is like we in Manipur, say if any hospital like we have RIMS and state government hospitals. So then if they want to start that genetic
I'll
lab,
help them. So you put me to the right person. I will help them to start from scratch, and I will visit there, see that it is done.
okay,
See,
it
your
really helpful.
research, if your research finding does not help the intended people, what point of doing the research? This is just to please
Yes, sir.
oneself, you know.
It is very nice of you, sir.
Yeah.
Actually, this is our that organization, PIMSA, is connected with various stakeholders in our state. So I think we should take the opportunity to start a lab in our state.
Will help you.
At the same time, we our new generation life science students to become genetic
Genetic
counselor.
counselor. Good, good.
Thank you, sir.
Thank you. Many of you are physicists, but not one of you are genetic counselor. I know at least 4 or 5 medical physicists from Manipur, but I don't know a single genetic counselor from, isn't it?
Yes, sir. The number of the physicists also like becoming very less. So for the
Acha,
last
okay.
10 years, no one had become. So
Okay.
we need to
Yeah.
we
Yeah.
need to promote our physics course. So
Yeah.
thank you.
Okay.
Yeah, so again, I could see hand raised by Kuraizam Sudeep. Yeah, Kuraizam Sudeep, please.
Yeah, first of all, yeah, this is Sudeep. I'm dialing in from US, and actually, I work for a sequencer company called Illumina.
Oh,
And
really?
yeah, if there's anything that I mean, I work on the cybersecurity part of things,
Yeah.
especially on the instruments as well as our
Yeah.
cloud and especially analysis, like tertiary analysis and more. There is a lot more in what you could do with that data as well. But besides that, first of all, thank you so much for your time and taking the time to really explain this. It's important for the population. But I'm just wondering from standpoint of public policy and what the institutions could do, perhaps for BRCA1 study in the population. Like an official study that could be sponsored, because at the individual level, it's quite expensive. Um, what is your what is your advice as to how to initiate such a study? That's one. Number two is that my understanding just from a very basic Google search is that it does there are other associated cancer risk as well, that has been talked about, whether that's even in pancreatic cancer or prostate cancer. There's a mild increase.
Yes, it is. I focused on breast and ovary, but prostate cancer around 15% and pancreatic cancer around 5%.
Right. Okay. So what would you advise about like from either a government sponsored or a certain initiative to do an actual local population study, an official one? How would one start go about doing that?
So it is important that it should be done. That's why I've said we have to find people who understand this cause, the importance of this, and and it may not be very expensive. To begin with, maybe just a study of say 1,000 or 2,000 people, irrespective of personal or family history of cancer. We get some idea, signal as to, because my starting point is only those who had breast and ovarian cancer who were referred for genetic testing. I'm finding very high number of them have the BRCA mutation and these two specific mutations. So once suppose if I after looking at these two specific mutation, and I can do it in just 1,000 rupees, these two specific mutation I can check for them in one individual whether they have this or they don't have this mutation. And if suppose we check in 1,000 people, so it will be just 1,000 people into 1,000 rupees. That makes how many? 10 lakhs, isn't it? So we can have a basic screen. And if we find something, then a larger population based studies, which should be done at least in 10, 20,000 people. And I do not know how homogeneous the Meitei community is in terms of the genetic makeup, or it is very diverse. I do not know. So are there some pockets of Meiteis who have sort of not married with the rest of the Meiteis because they may have a different mutation? Okay, I do not know. So based on all those factors, we can do that. And I think to begin with, the Meiteis who are outside, like in Delhi or Bombay or Bangalore, if we can just get, and sending the sample is very simple. We can test that, and once we get a signal, then and ultimately the local labs have to do it. The district hospital and the RIMS have to do this test. There's no point sending it to this thing, and this would not even require Illumina equipment. Just two specific point mutation. Like in Ashkenazi Jews, you just check for those specific point mutation. We can develop simple diagnostics. It will not be, you cannot do it at home, but your at least the district hospital or the medical college can do it easily.
I see. Just one last follow-up question, since you talked about the ethnicity. Basically, is there a way from just the patients that you had, like 75 patients, is there like is like a root mother gene that is kind of more common? Like, is it a certain lineage, like a pathogenic?
No. So it is all in BRCA1, and I've seen that none of them are related families. They are unrelated families. Okay, because otherwise you can trace. They will say okay, and from sometimes once or twice I have asked them also, are you related? No, they are not related to each other. So but obviously they have a common ancestor because all of them have the same mutation, and these are two different mutations. So maybe the exon 4 mutation is have a different common ancestor. So I've got two or maybe three different mutations in BRCA1 gene. So maybe three common, there will be three founders, two or three founders in the Meitei community who had first time this mutation, and from there it is coming down, down, down.
Right. Okay. Thank you.
Yeah. So before we come to the end of the session, I will pick up a last question, and with that we proceed with the ending of the session. So this is a question from a lady, TG Devi. Her question is, if breast tumor is benign, what is the chances that it will metastasize and become a cancer?
So benign breast tumors, or called fibroadenomas, they do not become cancers. Of course, very rarely anything can happen, but generally they do not become cancer. But the question is whether it is really benign or not. So depending on what age. So if suppose a 25-year-old lady has a lump in the breast which feels like a benign, as doctors we can, by examining the breast, how it feels and how it moves, we can suspect. And on ultrasound or mammography also it looks benign. We don't even do a biopsy for that if we are reasonably sure. If we have doubt, then we only will do a fine needle aspiration. But the same lump, any lump, breast lump in a lady over the age of 50, we have to rule out cancer, no matter what it looks like on feel or how it looks like on mammogram. Because as age increases, the cancer risk also increases.
Yeah. Thank you, Professor Sarin, and all the to the audiences present today for making this session very much exciting, as well as we are taking lots of information forward, which will be very much useful for us, as well as it can also be spreaded to people so that they are also aware of that.
Are there any oncologists in the group? I saw 1 or 2 names. Are there any cancer doctors here? No. Okay.
Yeah. So now let's proceed with the ending agenda. Now I request Dr. Brajagishwar, scientist, IBSD Imphal, Manipur, to offer the vote of thanks.
Good evening, everyone. Again, though it's already 9 p.m., it's my great pleasure to extend a heartfelt vote of thanks to our esteemed speaker, Professor Rajiv Sarin, MD, DNB, FRCR, Professor of Radiation Oncology, and in charge Genetic Unit, Cancer Genetics Unit, Tata Memorial Hospital, and Advanced Center for Treatment, Research, and Education in Cancer, ACTREC, Mumbai, for delivering such an insightful and thought-provoking lecture on BRCA1 and BRCA2 gene and their role in inherited cancer risks. You have not only illuminated the scientific basis behind BRCA1 mutation, but also emphasized the urgent need for awareness, early detection, and personalized approach to cancer prevention. Your talk has inspired us to think deeply about how genetic insights can be translated into better healthcare outcomes, especially in underrepresented communities like ours in the Northeast. On behalf of PIMSA, I express our sincere gratitude for your time, expertise, and patience. We are truly fortunate to have you had opportunity to learn you from today. And not only that, I also would like to invite you personally from IBSD, Department of Biotechnology, Government of India, for a visit to Imphal, so that we can have a large audience and give lecture to almost to all our PhD students, all our cancer research students. So I think that will be that will be very much beneficial to all. And I thank you once again, and we hope to welcome you back in the future for more enriching session. And I also would like to thank my sincere thanks to President of PIMSA, Dr. Ranjan Kumar Khaidem, Rina Fry Lapam, PIMSA Manager Heramani, Dr. Romi, moderator of today's session, for managing and arranging this lecture session a success. And thank you, everyone. Jai Hind. Dr. Romi, please.